In this chapter, we're exploring population and evolutionary genetics. We are exploring the genetic differences between individuals, how that we know how that uh, variation among individuals arises through the process of mutation, but it's thinking about this in the context of how that creates phenotypic variation within a species, and then at a much deeper time scale, how that can lead to the origin of different species. We're also going to touch briefly on how you can use genetic information to reconstruct that evolutionary history and explore processes within populations. So, the first thing we need to do is recognize that species are variable. We see that when we're sitting in a classroom. You see it when you're sitting at your dining room table with your family. None of you are identical, none of us are identical, and that is all due to the variation introduced into our genomes through the process of mutation. Okay? And the differences between us and other species is also the product of mutation, but it's mutation that has happened over very long periods of time. So it's important that we enter this chapter recognizing this abundance of variation within populations, and note that populations aren't fixed. They don't exist in some optimal genetic state. Okay, and by that I mean the variation exists at the level of alleles. So there are lots of alleles for most all genes within a species. And though the process of mutation introduces new genetic variation, we lose genetic variation in populations as well. We lose it through the process of selection and drift that we're going to get into. Now, you've heard of natural selection before, and you probably think of it in the context of the common phrase survival of the fittest. So let's imagine a population of cheetahs. Some cheetahs are faster than others. The slowest of the cheetahs starve, right? The fittest of the cheetahs that are fast enough are able to eat, they survive, and they have offspring. What you expect over time is that population will lose the alleles that cause cheetahs to be slow and hungry and starve because they don't live, they don't reproduce, they don't have slow, hungry offspring. And that is, in fact, the case, that those alleles that are maladaptive, that are bad for you, they do tend to be removed from the population relatively quickly. But new mutations are introduced into the population constantly. Some of those are good, but not many, as we've talked about. And many of those have no effect at all. And those that do have an effect, most of them are bad. Okay? So, in the context of thinking about evolutionary genetics, I want to briefly introduce to you the fathers, plural, of the theory of evolution, and that is Charles Darwin, who is quite famous for taking a circumglobal trip on the HMS Beagle over a 5-year period where he visited a number of localities, most famously the Galapagos Islands off the northwest coast of South America, but also importantly, many other points along the mainland coast in South America, where he made a lot of stunning observations about biodiversity that really helped him formulate his idea of evolution by natural selection. Though he got back to England in 1836, it took him a long time to kind of figure out what he had seen and synthesize his thoughts with respect to the change in organisms over time. But he did read an essay by an economist named Thomas Malthus, and that really helped him understand how populations change over time. And once he recognized this mechanism, he worked for a long time to try to get things in place so that he could present this radical new idea to the world. Now, the essay that Malthus wrote was basically natural selection in humans. He was writing about the incredible human population growth taking place in England. And what he said was, there are going to be so many people born that we are going to not be able to feed all of them. So more offspring are going to be produced than can survive. But what Malthus pointed out was who survives and who doesn't is not going to be random. Those that survive are going to be the ones that have beneficial traits. In this case, the beneficial trait would be family money. If your family's got money, you're going to survive when food supplies are low because you can pay more for it. If you're poor, food supplies get short, then you are more likely to starve. And what Darwin saw in that was a mechanism for determining who lives and who dies in the natural world. And in our cheetah example, it's the slow cheetahs that don't survive and the fast cheetahs that do. Okay, it's not random. But in 1858, 22 years after he returns, he gets an essay from a young naturalist, Alfred Russel Wallace. This guy was quite amazing. He spent a great deal of time in the neotropics in the Amazon basin, and then also in Southeast Asia. And unlike Darwin, he wasn't a rich guy that didn't really ever have to work and could sit and think about things. He had to go out into the jungle and collect animals, and he sold these to collectors in England. And not living animals, but mounted insects, mounted birds, and these were the things that he was collecting constantly. And it's his observations of the differences among populations and how these things are so similar to one another, but have changed ever so slightly depending on the conditions of the island that they're on, for example, that helped him formulate his same idea of how populations change over time in response to what we call natural selection. So this theory of evolution that they came up with includes the idea that organisms change over time, their characteristics change over time. It also is based upon the idea that these living organisms share a common ancestor. So when Wallace was on two different islands and saw similar butterflies on those two islands, though they were not identical, his hypothesis was that those two similar butterflies are on such proximate islands because there was some common ancestor in the region from which those two are descended. Okay. They also suggest that change, evolutionary change, is quite gradual, and this is quite consistent with what we see. And then it's all about populations and proportions. And what I mean by this is that the variation that exists within populations and the change in those populations over time has everything to do with allele frequencies and genotype frequencies and the change in those frequencies over time. Okay? The variation in the cheetah population that I've described exists in the form of different speeds for different individuals. Those different speeds being determined by the genotypes those individuals have, which are, of course, combinations of alleles that they possess, those different alleles existing because of mutation. And then natural selection, of course, is the idea that the forces of the environment in which an organism exists exert pressures that non-randomly determine or contribute to survival rates for the different genotypes that exist within a population. Darwin and Wallace were not familiar with the work of Gregor Mendel, but with their hypotheses and the understanding of patterns of heritability that Mendel put together, the following synthesis after their discoveries, their respective discoveries, led to a more comprehensive understanding of how organisms change over time. Now, a term that we're going to touch on here in this chapter is population genetics. And in this context, a population is a definable group of individuals of the same species. And so these individuals exist as members of the same species and may potentially interbreed with one another, produce offspring um, that have genotypes that are derived from both parents that exist within this population. Now, what we've thought about so far in the context of different alleles and genotypes is particular pairings of one mother and one father and the alleles that those two individuals have, and using a Punnett square to determine what offspring they might have and what the relative frequencies or ratios of those offspring genotypes might be. Now, in the context of a population, it operates in much the same way. The same alleles are the things that we consider. Their frequencies are the important metrics by which we understand the genetic composition of the population. And so collectively, all of the alleles of individuals within the population are the gene pool for that population. So you can think of the gene pool for human eye color being not just let's say there are X number of alleles for eye color. It's not X number of alleles. The gene pool is there are 330 million of us in the United States, let's say. Each of us has 2 copies of that gene, okay? That means there are 660 copies of the gene in the United States, 660 million copies of the gene in the United States. Of that, some proportion are the allele for brown eyes, some proportion are the allele for green eyes, blue eyes, so on and so forth. That is the gene pool of our population, this pool of 660 million copies of the gene that determines eye color, okay? So when we start thinking about the frequency of an allele in the population, let's say the allele that confers brown eyes, we need to think about the things that contribute or affect allele frequencies over time, okay? Right now, the allele frequency for the brown-eyed allele in the human population in the United States, it is what it is. It's a number. We could go around and count the frequency of that allele. It would be the number divided by 660 million, right? That's the frequency of that allele in our population right now. But from a evolutionary genetic perspective, we're not interested in necessarily what it is right now. We're thinking about how does that allele frequency change? That the frequency of that allele might increase over time or it might decrease over time. The things that contribute to the change in allele frequencies are listed here, okay? Allele frequencies change due to the process of mutation, okay? They change through migration and they change through genetic drift, okay? And they also change through natural selection, as I've already described. Now, we've got lots of genetic variation within all populations, including our own. And you saw this when you lined up DNA sequences, or you will see it if you haven't done Lab 7 yet. When you align DNA sequences from different individuals or different species, here I'm showing you the 2 human individuals, the Somalian and the Tibetan, lining up their homologous gene sequences. And you can see that there are 2 different alleles here where these 2 individuals differ in one particular nucleotide, okay? But there are many more than 2 alleles for any particular locus in a population, quite frequently. And many conditions that we see, many phenotypic traits, might be due to mutations at very different gene regions. This came up in the context of the different types of cancers that might have the same name, all be called colon cancer, but they might be due to different mutations. In humans, the genetic disease cystic fibrosis, there are more than 1,500 mutations, in many of which are indicated here in this particular gene, and their relative abundance indicated by the height of the black bar above that portion of the gene. But there are many different mutations that can result in this cystic fibrosis phenotype in the population. Okay? So there is lots of genetic diversity within populations. That genetic diversity exists in the form of alleles. The frequency of these alleles changes over time by random chance. That's drift. We're going to talk about that. Due to mutation, due to migration. We'll go into detail about how migration affects frequency, and then also through the process of natural selection. Now, first, we're going to think about the frequency of alleles and genotype in the absence of any of those things that affect it. Okay? We're simply going to talk about the frequency of alleles and phenotypes within a population to begin with. Now here you see an example of albino organisms, a squirrel, a tiger, a peacock, and a python. Each of these individuals that you see, the white ones, have a mutation that reduces their or prevents them from producing melanin. So they don't have any dark pigment as you would normally see. Now, in almost all cases, this lack of pigment is a recessive mutation. And so these individuals that are white must be homozygous recessive. And the individuals that have normal coloration are going to be homozygous dominant or heterozygous. Notice that there are two different phenotypes here, but there are three different genotypes, two of which provide the same phenotype. Okay? So we're going to talk a lot about allele frequency and genotype frequency in this chapter. So let's just use an example with the squirrels here real quickly. If there are 100 squirrels in the population on the campus and 2 of them are albino, okay, I've given you some information there, and I've given you a frequency. I've only given you one frequency. If we go back to the previous slide, there are 5 different things I've shown you there. 2 phenotypes, 3 genotypes. What did I give you here? I've given you the frequency of the albino phenotype in the population. And since there is only one genotype that can produce that phenotype, I've also given you the frequency of that genotype, the homozygous recessive genotype. So there are 98 other squirrels in the population, and if I tell you that 30 of those carry the allele, they're heterozygous, we've got another frequency, another genotype frequency. So what I've given you here is the genotype frequency of the homozygous recessive genotype is 2%. The frequency of the heterozygous genotype is 30%. Okay, I can already look at this, and how many other genotypes are there? There's only one other genotype, right? The homozygous dominant. You can already tell me what the frequency of that homozygous dominant genotype is by looking at this. Because there are only 3 different genotypes, and their frequency has to sum to 100%, then the frequency of the homozygous dominant genotype here is 100% minus 32%. So 68% of the population are homozygous dominant. Let's think about what this means at the level of the allele, though, okay? Because within this population of 100 squirrels, since they are diploid, there are 200 copies of this gene in the population. Remember, we are using big A or little a for the alleles of this gene. So you can think of it as there are 200 copies of A in the population. Each of them is either the dominant A or the recessive A, but there are 200 copies of the gene. Of those, 4 copies exist in those 2 albinos. Remember, because they are homozygous recessive. Each of them has 2 copies of the recessive allele. Those 30 individuals that are heterozygous, they have 1 big A and 1 little a. So those 30 individuals have together 30 copies of the recessive allele. So out of all 200 copies in the population, 34 of them are 17% are the recessive allele. So 17% of the copies of the gene in the population are the little a, or the recessive A. And you can do the math from there. 83% of the copies of the gene in the population are the dominant allele. So these are 5 frequencies we've just calculated. 2% is the homozygous recessive genotype frequency. 30% is the heterozygous genotype frequency. 68% is the homozygous dominant genotype frequency. 17% is the recessive allele frequency, and then 83% is the dominant allele frequency. Okay? The 3 genotype frequencies have to add up to 1 or 100%. The 2 allele frequencies have to add up to 1 or 100%. Okay? So using these frequencies to any one of these to calculate the others is something that you're going to need to be familiar with. And I'm going to walk through how we do that here. Okay? So this is where we revisit something that you've seen before, the Hardy-Weinberg law, in which we take each allele for a locus and assign it some letter that represents its frequency. You've most frequently seen P and Q, and so that's what we're going to use. Okay? And I'm going to use a scenario where we're talking about gene R, where there's a dominant big R, a recessive little r, and we might assign P to represent the frequency of the dominant R, and Q to represent the frequency of the little r. Okay? So we have 2 alleles, each exists at some frequency. The frequency of those 2 alleles has to sum to 1 or 100%. Under Hardy-Weinberg predictions, the frequency of these alleles allows us to use the product rule of probability to predict the frequencies of genotypes. Okay? So you've seen this equation below, p squared plus 2pq plus q squared equals 1. Okay? And this is the equation that we use to represent the genotypes. And I don't know how many times I have taught the Hardy-Weinberg equation, and people are not able to explain that each of these 3 components of the Hardy-Weinberg equation represents a genotype. P squared, because we equated P with the frequency of the dominant R, P squared equates to the frequency of the homozygous dominant genotype. Q squared equates to, in having taught Hardy-Weinberg a number of times, I'm continuously amazed by the difficulty students have reconciling exactly what each of these components of the Hardy-Weinberg equation represents. And so I want to make very clear here that because we have assigned the allele big R the letter P to represent its frequency and the letter Q to represent the frequency of the recessive R, that each of these components of the Hardy-Weinberg equation specifically reflect the frequency of a genotype. So P squared represents the predicted frequency of the homozygous dominant genotype. Q squared represents the expected frequency of the homozygous recessive genotype. And 2PQ represents the frequency of the heterozygote. Okay? And what we are seeing here is another application of the product rule of probability. Okay? Because I want you to think about all the Punnett squares you've ever seen. Okay? And when you think about the giant gene pool that exists for any organism in which there are 2 alleles for a particular gene, in this case R. There are lots of big R's in the population, there are lots of little r's in the population. If I ask you to tell me what is the probability that an individual is born homozygous dominant, you can do that using this in the product rule of probability as you understand it. So if the frequency of big R is 0.6, 60% of the copies of that gene in the population are big R. I can tell you that the frequency of big R in the population is 0.6. What is the probability that a baby is born that is homozygous dominant, homozygous for big R? Well, it is the probability of getting an egg that has a big R and the probability of getting a sperm that has a big R. Well, when we think about that egg and sperm, unlike when we did Punnett squares, we're thinking of all the eggs in the population. We're thinking of all the sperm in the population. If 60% of the copies of this gene in the population are the dominant R, then that means 60% of the eggs are going to have the dominant R and 60% of the sperm are going to have the dominant R. What's the probability of getting a dominant R containing sperm? 60%. What's the probability of getting a dominant R containing egg? Also 60%. So the probability of both of those things happening to create this one individual, we use the product rule, 0.6 times 0.6. I expect, given that, 36% of the population will be homozygous dominant. I expect a 36% chance that any individual born is homozygous dominant. Okay? This math is really simple, and I'm going to go through it again here in a moment in a way that looks a lot like a Punnett square, but it's critical that you understand what each of these components of the Hardy-Weinberg equation represent. Now, the Hardy-Weinberg makes some assumptions. It is effectively a null hypothesis in which we assume that the population is really large. We assume that all individuals are randomly mating with one another. We're assuming, let's say, that albino squirrels and brown squirrels aren't mating separately, that they're just as likely to mate with an opposite colored squirrel as the same colored squirrel. We assume there's no migration, no selection, no mutation to this allele in the population. All of these assumptions of the Hardy-Weinberg law are the basis for evolutionary change in populations. Meaning, if none of this happens, the population never changes. Okay? Allele frequencies will stay exactly the same. And if they are going to stay exactly the same, we can just use the current allele frequencies to predict genotype frequencies in the future. Okay? If we find, the reason it's a null hypothesis is because we use it to determine whether we find that our observed genotype frequencies are different than we would expect. They would be different than we would expect if one of these things is happening. If the population's too small, if the mating is not random, if there's lots of migration, if there's lots of selection, if there's mutation to this gene, okay? All of those things would, if those things are happening, they would cause the Hardy-Weinberg equation to not accurately reflect or predict genotype frequencies, okay? Now let's look at this example with sperm and egg using allele frequencies to predict genotype frequencies. This looks complicated, okay? But I want you to recognize that this is exactly what you did with a Punnett square, only if you notice here, the frequency of these two alleles, big A and little a, in the population is 0.7 and 0.3 respectively. So 70% of the copies of the gene in the population are the dominant A, 30% are the recessive A. And that means 70% of the sperm will have the dominant A, 30% of the sperm will have the recessive A. Same with the eggs, 70 and 30%. When you did a Punnett square with one father and one mother, there were 2 columns and 2 rows, just like this. The only difference was each of the alleles in the columns and each of the alleles in the rows had a 50% chance, right? That parent had 2 different copies, or 2 copies of the gene, and each one was equally likely to be in the sperm and the egg, all right? This is different because we're not talking about an individual where it's a 50-50 chance. We're talking about a whole population where it's a 70-30 chance that there's a big A in the sperm or a big A in the eggs. Now, we used this same product rule to calculate the genotype frequency expectations for Punnett squares with individual parents. But here the numbers are not 25, 25, 25, 25. Okay? The probability of putting two big A's together, a sperm and an egg, is 0.49. Okay, 7 times 0.7 times 0.7. The probability of putting two little A's together in a zygote is 0.3 times 0.3, so 9%. And then the probability of a big A egg and a little a sperm is 0.21. Probability of a big A sperm and a little a egg is also 0.21. But note that those two, the top right, the bottom left, the blue squares, are the exact same genotype. They're heterozygotes. There are two different ways to make a heterozygote. That is why in the Hardy-Weinberg equation, to calculate the homozygous dominant genotype frequency, top left, we just use p squared. P being the frequency of A, 0.7. 0.7 times 0.7 squared, so 0.7 squared is 0.49. Q squared is the frequency of the homozygous recessive genotype here, 0.3 squared, 9%. And then the frequency of the heterozygotes, that's the center part of the Hardy-Weinberg equation, 2pq. In the top right blue box, you see P times Q, 0.7 times 0.3. In the bottom left box, you see Q times P, 0.3 times 0.7. Because those are both the same genotype, that central portion of the Hardy-Weinberg equation that is meant to predict the frequency of heterozygotes is 2 times P times Q. Because in each of these blue squares we have P times Q. You double it, you get the frequency of heterozygotes. In this case, it's 0.42. Got it? So this is how we get the P squared, the Q squared, and the 2pq. Okay, this is illustrating this combination of alleles that happens at the population level. Remember, this is not an individual Punnett square. Now, Hardy-Weinberg is a prediction that we would expect to see in situations, in populations. We expect populations to match or fit the Hardy-Weinberg if there's no selection or change taking place in the population. What's interesting is that even in large populations like ours, we can see things that change in frequency. Human populations are subject to selection, and so we could either be existing at some steady, stable state in Hardy-Weinberg equilibrium for a particular locus, or we could be under very strong selection favoring a particular locus, violating the Hardy-Weinberg expectations, resulting in genotype frequencies that differ from what we would expect. A great example that your text digs into is one that I hope you will find interesting. It's quite a remarkable discovery, the role of this gene, in that there is a variant of this gene that exists within the human population. Okay, this gene is CCR5. It has 4 exons, and in some individuals, there is a 32 nucleotide deletion in the 4th exon. Okay, and that allele is just called delta 32. Okay, so there's a normal allele where it's the proper length, and then there is a shorter 32 nucleotide shorter allele in the human population. Now, if these were just different alleles and they had no effect on our fitness, there weren't lots of mutations to it, the frequency of this in the human population would probably stay stable over time. But what's fascinating is that this allele confers resistance to HIV infection. So if an individual possesses this delta 32 mutation, they are resistant to HIV infection. Well, that sounds great. So is it going to be subject to selection and increase in frequency in the human population? Well, it's quite uncommon globally. It's most common in Scandinavian countries, and this has been hinted as being a consequence of individuals with this mutation having a greater chance of surviving early plagues that affected Europe. But regardless of where it started and how it became this abundant in this region, you can see that the allele frequency is still relatively low, lower than 20% of the copies of this gene, even in its most common area, are still quite low. If we look at a gel electrophoresis of individuals with the different genotypes, on the left, either homozygous for the wild type, on the right, heterozygous, or on the left, homozygous for the delta 32 allele, we can see that if you take this locus and cut it in two pieces, the CCR5 gene, you cut it into two pieces, okay? If you have the wild type, you're going to get a piece that is 332 bases long and 403 bases long. So you just take one long 735 base fragment and cut it into two pieces. That's what you're going to get, okay? If you have an individual that's homozygous for this delta 32 allele, you cut it with this restriction enzyme in a particular spot. You still have one fragment that's 332 bases, but your other fragment, instead of being 403, is 32 bases shorter. So you can see that genotype is homozygous recessive. They only have the shorter portion of that 403 base fragment. The heterozygous individual has both a 403 band and a 371 band. So this should make clear to you, I hope. If not, take time to think about it. You know how gel electrophoresis works. This illustrates the size difference in this gene region for individuals with the delta 32 allele. And now there's even a table presented in your text that you can calculate the genotype and allele frequencies for the 1 and the delta 32 allele in the human population and see if it fits your Hardy-Weinberg expectations, okay? Now, turns out that the human population is so large that this is not under strong selection in the human population, and it's not changing in frequency all that much. But it's an interesting exercise to explore the frequency of these genotypes and alleles within the human population. So on the exam, you will definitely be asked to make some calculations of frequencies. And I want you to recognize that in these types of calculations that we're discussing in this chapter, there are 5 numbers that are calculable or available, some of which I will give you, some of which you have to calculate. There are going to be 2 allele frequencies and 3 genotype frequencies. Make sure you understand what's what I give you and what you need to calculate. Here's an example. And I want you to work through the examples I give you. I am not giving you the answers to these in this lecture, okay? I want you to use the information I've just lectured on to work through these and calculate them, and visit with me during office hours to go over all of these calculations and ensure that you've done it correctly. So in this first example, I am giving you the frequency of one of the alleles, the dominant G, okay? I then ask you what's the frequency of the other allele, and then the frequency of the 3 genotypes. Remember, there are 5 numbers here, and I gave you one of them. You should be able to use that and the Hardy-Weinberg equation to calculate the other 4. In this example, I'm telling you that a population is in Hardy-Weinberg equilibrium. The one number I'm giving you is a genotype frequency. And what you should be able to do is use the Hardy-Weinberg equation to take that genotype frequency and figure out the other 4 numbers. Those 4 being the other 2 genotype frequencies and the 2 allele frequencies. I clearly made a typo here. I don't want you to calculate the heterozygous genotype frequency twice. I want you to calculate the frequency of heterozygotes and homozygous recessive individuals. I apologize for the typo. Okay, now if you're to calculate the frequencies, genotype and or allele frequencies for a locus that has more than 2 alleles, because as I've made clear to you, there are more than 2 alleles for nearly all genes, okay? And the ABO blood type system is one of these examples that I've given. You use the same principle. You apply a letter to represent the frequency of the allele. So in this case, we might make P the frequency of the A allele, Q, the frequency of the B allele, and R, the frequency of the O allele. I'm not going to give you one of these on the exam, but just know that I could use the same principle we applied in a 2 allele system with P squared plus 2PQ plus Q squared equals 1 to this system in which there are 3 alleles. Okay? Now necessarily the frequency of these 3 alleles has to add up to 1, but so too does the frequency of all the genotypes that are possible. What are the genotypes that are possible? You can be homozygous for A, homozygous for B, or homozygous for O. That would be P squared, Q squared, and R squared respectively. Okay? Now what other genotypes are possible? AB, AO, BO is also possible. So we can have A with B, A with O, we can have B with O. So those are 3 heterozygote genotypes that would be reflected in the Hardy-Weinberg equation by 2PQ, the other would be 2QR, and the other would be 2PR. Okay? That is all possible heterozygous genotypes. So instead of the Hardy-Weinberg equation having 3 components, it's going to have 6. But again, they still have to add up to 1. 100% of the genotypes are represented there, and their frequencies add up to 100%, or 1.0. Okay, so now we start thinking about the process of natural selection and how this might change allele and genotype frequencies within a population. Okay, this is probably the most prominent force that affects the frequency of alleles within populations. So because this variation exists within populations and it's passed from parent to offspring, and there are more offspring produced than can survive, this was the problem that Thomas Malthus recognized in the English countryside, and that individuals that are better suited to survival do survive. That means because those individuals are better suited and those traits that make them better suited are passed on genetically, it's heritable, they have a better chance of surviving, they have more offspring, their offspring are more like them, the population becomes more like them. Okay, the population is better suited to survival. So this is this fundamental principle of evolution, that these heritable traits, through the process of selection, will see the frequency of alleles in populations change. Okay? Now recognize that selection can be both natural and artificial. Natural selection being, basically all the forces that are affecting the squirrels out in the grove today. Whereas artificial selection can see populations change over time through the selective breeding that we do. That's why we have produced through selective breeding things like Great Danes and Chihuahuas, even though they're the same species. We have created separate population pools that we decide, we select what traits, what individuals are suitable for moving on to the next generation. We don't necessarily remove individuals from the population in the same way that slow cheetahs are removed from the population or blind squirrels are removed from the population. But we simply decide not to breed the smallest of the Great Danes or the largest of the Chihuahuas. Okay? And so this gets at something that you've heard about before, almost certainly, but probably don't have a good grasp of, and that is fitness. And when we use the term fitness in a biological context, it means one and only one thing. It means basically, how good are you at passing your alleles on to the next generation and the generation after that? Okay? So it is really all about the production of offspring and your offspring being successful in reproducing and producing their own offspring. That is fitness. Okay? Now, if the population is under this selection that is removing alleles from the population that make you less adept at surviving and or thriving, then why aren't all those bad mutations, those deleterious alleles removed from the population? Well, there's a great figure in the text that looks at the frequency of an allele, this lowercase a, that is lethal. So this is a recessive allele that is lethal. So if you're homozygous for this, you're dead. Okay? You would think that this would be the most rapidly removed allele from a population. But I want you to look at this graph. Look how rapidly, or slowly actually, this recessive A allele decreases in frequency from an initial frequency of 50%. What that means is that half of the individuals in the population, half of the allele copies in the population are this recessive A that is lethal in homozygotes. If you do the Hardy-Weinberg equation with the recessive lethal allele at a frequency of 0.5, that means 25%, right? Q squared. 25% of those individuals that are born in the next generation are going to be homozygous and die. And so that's removing this recessive allele from the population at a much higher rate than the dominant allele. And so the frequency of this allele is going to decrease quite dramatically at the beginning. Look at how much it changes from generation 1 to generation 2. Okay? But look how much more slowly it decreases in frequency to generation 100. It takes 100 generations to remove this allele from the population. And this is the worst possible kind of allele you could have. It's lethal. The reason it is so slow is that the rarer this allele gets, when it's below 10%, when it's at 5% frequency, remember what has to happen for this allele to actually cause the individual bearing it to suffer. You would have to be homozygous for it in order to die, okay? But if the frequency of this allele gets below, say, 5 or 10%, what's the frequency of individuals that are homozygous for it? It's incredibly low. Q squared, when the frequency is 5%, is so low. You square 0.05, that is a very small number. That means 2%, 2.5% of the population are going to die because they're homozygous recessive. It's so rare that it does not affect a significant number of individuals in the population, and so it's slow to leave, okay? So deleterious mutations pop up all the time, and when they're bad, it takes a while to get rid of them, especially when they're rare, okay? Now, the strength of selection on an allele or against an allele has everything to do with how fast it disappears from the population. So we just showed or discussed an example that matches pretty much this red line to the far left, that if it is a lethal allele, if it starts out at a frequency of 1, this lethal allele is going to disappear incredibly fast, okay? So the strength of selection against this recessive A allele is super high. It's going to disappear, but it's still going to take nearly 100 generations. Most alleles do not have that negative of an effect on the individual that bears them, and so it would take much longer to remove that allele from the population. You can see that if the difference, the selection strength against the allele is low, it might persist for millions of years, not just hundreds of generations. Okay, so we're going to talk about how selection acts on populations. And the first thing I want to show you is an example of the most commonly observed and the most intuitive form of selection, and that is directional selection. And the example that I'm going to use and your textbook uses is one of the depth of the beaks of Darwin's finches on the Galapagos Islands. So this is a remarkable group of finches that inhabits the islands of the Galapagos off the western coast of South America. And what's interesting is there's lots of diversity in these species, and they eat a lot of different things. This particular medium ground finch, that's literally the name, very descriptive. This medium ground finch eats seeds, okay? And some seeds are larger than others. The deeper the beak of the finch, the larger the seeds it can consume. And what you'll see here in this graph is this really interesting observation that some researchers made over a few decades of observation. And that is that the phenotype of the population, their beak depth, changed over time in response to changes in the environment. So in years when it was dry, what they saw was that the size of the finch's beaks increased quite dramatically. Okay? So this increase in beak size is due to the ability of those individuals with larger beaks to survive when there's a drought because they can eat larger things that they wouldn't normally eat. So you have to recognize what's happening here is that the finch's beaks are not getting larger because they're looking around trying to find stuff to eat and they're getting hungry and their beaks just get bigger so they can eat the harder seeds. What is happening is in the population, some individuals have big beaks. Some have medium, some have small beaks. What happens in these years when it's dry is that the average beak size increases because all the individuals with the small beaks starve to death. And so the big ones had a higher chance of survival. They had more offspring. In the next generation, there's a lot of individuals with bigger beaks because it's heritable variation. Let's look at why we call it directional selection. It is taking the distribution of this trait value in the population and pushing it in one direction. I want you to look first at this dashed line. What this is a normal distribution. It's a curve for how large, how deep is the beak of every finch on the island. And imagine the x-axis is how big is your beak. Far to the left is the smallest beaked individual. Far to the right is the largest beaked individual. Okay? Somewhere in the middle, there are lots of average middle depth beaked individuals. The y-axis is frequency. So the higher that point in the curve, the more individuals there are with that beak height on the x-axis. Okay? Now what we're seeing from the dashed line to the green line is a shift in the population. Look at that dashed line right now. Where is the average beak depth? It's right at the peak of that normal distribution. The hump is pretty much the average. But look at the hump in the green distribution. Okay? After selection, the population has a larger average. Okay? Look at the very far right point of the green curve. That is the thickest beaked individual in the population. His is thicker than anybody was in the previous generation. The average has increased, the largest beak has increased, and look, even the smallest beak that exists in the population after selection is significantly larger than the smallest beak before. All those individuals that are not in the overlap region, they were left out. Okay, the vast majority of those small beaked individuals did not survive. So who did? Think of those individuals in the area where the two graphs overlap. Those individuals survived and produced, on average, larger beaked offspring. What has happened is the average has been pushed in one direction. There's been a shift in the average. The whole distribution has shifted in one direction. Okay? That is why we call this directional selection. Contrast this with stabilizing selection, where instead of the selection pushing the distribution in one direction or the next or the other, because one end of the continuum, one of the extremes, is really bad. In the last example, it was really bad to have a small beak. It was great to have a big beak. So everything shifted towards larger beaks. In stabilizing selection, what we have is the optimal state is no longer one of the ends of the distribution as it was with the beaks. It's the middle. Stabilizing selection is a situation where the average is the best. If you are an outlier to the far right, to the far left, that is bad. Okay? And so selection acts against both extremes. And so what you end up with is a tightening or a consolidation around the average, because that is the best trait value for this population. In this example, we're looking at human birth weight. And so we see the frequency of birth weights in humans. So the red line corresponds with the right side y-axis. So the percent of births in the population that are 7 pounds or 6.5 pounds is right about 20%. That's that peak, okay? A very small percent of the births in the population are 10 pounds. A very small percent are 2 pounds, okay? But we have a lot of births in that middle region. Now look at the left side y-axis, the percent mortality and birth weight, okay? There are many more deaths at the low end of the spectrum than there are at the average birth weight. There are a fair number more deaths at the far right end, the heavier end of the spectrum, than there are at the average. You see there's much higher mortality at the low end and the high end than there is right in the middle at the average. And so what happens is selection is picking against big and small and consolidating the population, pushing it towards this average and maintaining that stable state. The last type of selection is disruptive, in which you take a population. This is an experimental population that was selected for 2 different trait extremes. So these fruit flies, which if you look at a zoomed in picture of a fly, they have lots of hairs on their body. In this population, one population, they let breed the individuals with the most hair and the fewest hair, and nothing in between. Every generation they took the hairiest and the least hairy and bred them together. And so that was favoring both ends or both extremes and disfavoring the average. So it's kind of the opposite or the inverse of stabilizing selection. It's not like directional selection either, because in directional, only one of the extremes is selected against. Okay? Here, both of them are selected for. Okay, I think the easiest way to think about it is all 3 of them side by side, and I found this image online that indicates for all 3 the population before and after selection, and you can see what happens. In directional selection, it's easy. The average moves to the right. Stabilizing selection, I think this illustrates the principle much better in that the height of the peak after gets higher, but the sides consolidate, they pull in, they tighten up around the medium, the average trait value. And in disruptive, it breaks it up and favors, and the extremes become more frequent. The 3 examples here are quite silly, but they're very illustrative, right? Directional selection, you go from having a small tail to a larger tail, right? It just pushes the population towards having a larger tail. In stabilizing, having a really tiny tail sucks, having a ridiculously long tail sucks, but right in the middle is amazing, and selection favors that and keeps tail length right there in the middle. In disruptive selection, you end up with selection against both, or against average length tails. So it's good to have either a really long tail or a really short tail, okay? So this is a great example. They use actually ground squirrels and tree squirrels, where the different types of tails are very useful in the different environments in which those types of squirrels live. So ground squirrels, you probably know as chipmunks, they have very little tail to think about, whereas tree squirrels have very long tails. And so selection has pushed some squirrels to having a larger tail and some to having a shorter tail. Okay, so we're talking about allele frequencies changing in populations, and spent most of that time talking about selection. Okay, selection definitely affects the frequency of the alleles responsible for, say, tail length, okay, or human birth weight. Now, there are a couple other things that can change the frequency of alleles within populations, and it's important we consider those. The first is mutation, and then we'll talk about migration and drift. So the mutation rate is a very small player here, okay? If you think about the frequency of the allele for green eyes in the human population, That frequency is pretty low, but it's out of many billion people, right? And if when you produce the egg or the sperm that becomes your first child, you make a mistake and though you don't have the green-eyed allele, you give to them a mutated version of the eye color gene that gives them a green-eyed allele. That mutation is not noticeably or perceptibly changing the frequency of the green-eyed allele in the human population. This is the way in which, through mutation, we get all mutations. All genetic variation happens through mutations, but it's not a huge driver of changes in the frequency of alleles. And this figure from your text makes that quite clear. Okay, so the next thing that we're going to talk about is migration and how migration affects the frequency of alleles. All right, this is a complicated looking equation, but I've broken it down a little bit and provided some explanation down here in the bottom that will help you understand what's going on, okay? And so here, when we talk about the frequency of these different alleles in the population, we need to think about how the movement of individuals from one population to the next or to another affects allele frequencies. And it only does this if the two populations have different allele frequencies to begin with, okay? So here, just think of P the way we've used it in the Hardy-Weinberg equilibrium. It's the frequency of one of the alleles, P or Q, right? So this is P. So in this, I have two different I's, and instead of M in your text, I use the letter C. So we are going to consider a scenario where there's a mainland source and an island, okay? So there's a mainland and an island, each inhabited by this species. But the allele frequencies are different in these different places, okay? So in this equation, you have this italicized M, which represents the number of migrants, or the proportion of the individuals that are breeding on the island that came from the mainland, okay? And then, so that's what M means. We're interested in this island. I'm going to show you the island in a minute. But the M represents the proportion of individuals breeding on the island that came from the mainland. The PI, PC, and PI prime, those three things represent the frequency of the dominant allele, okay? So the frequency of the allele on the coast is different from the allele frequency on the island. And what we're going to calculate is PI prime. What's the frequency on the island going to be in the next generation if some proportion of the individuals that breed now came from the mainland instead of the island? So you should be able to apply this equation to the example on the next slide, okay? But you're only going to do it for one allele. You don't need to do it for both alleles in this equation. Once you know the frequency of one allele, you can figure out the other. So if you apply that equation to this scenario where we have two different populations, some on the mainland, some on the island, and the allele frequencies are quite different, okay? You can see the allele frequencies here. If I tell you that 10% of the individuals that breed on the island this year come from the mainland, that's going to change the allele frequency of the island, okay? Look at the frequency of the little d on the island, 0.4. Contrast that with the frequency on the mainland, which is 0.7. If 10% of the individuals that breed on the island this year bring that higher frequency of recessive D to the island, you're going to have, it's not going to be 0.4 next year, it's going to be higher, okay? So you can use that equation on the previous slide to run this calculation. And in this case, M, that italicized M, is 0.1, right? I said here 10%. So 90% of the individuals that breed on the island bring with them a frequency of D of 0.4, little d 0.4, and 10% of the individuals bring a allelic frequency of 0.7. Okay? So this is an example, and if you run through these calculations, you will see how the frequency of the recessive allele changes on the island. Okay? So 90% of the individuals come from the island that breed, 10% come from the mainland. So we can see allele frequencies change, and we can see the historical signature of this change even in human populations. This is the frequency of the B allele for human blood type, and this is an allele that originates in Asia, but the frequency of it is more common than you would expect in Europe, and it kind of, you can see there's a very dramatic gradient as you go from Asia into Europe that is reflective of the history of migration of people from Asia into the European region. All right, so now that we've covered mutation, migration, and selection, the last thing we need to touch on is genetic drift. And drift is probably one of the more complicated aspects of changes in allele frequencies that I've got to convey to you. And really what it is, it's described as the random walk. It's random change in allele frequency. Remember how we took allele frequencies and used them to predict genotype frequencies in the next generation with the Hardy-Weinberg equation? Well, that's what we expect if everything's the same, but things don't always work out that way. Our predictions are not always 100%, and the next generation could be biased. There could be a few more homozygous dominant individuals than we expected or predicted. There could be a couple fewer homozygous recessive individuals, and that changes the landscape for the subsequent generations because there are more of one than we had at the beginning. So here is a illustration of a computer simulation of a population that was started with an allele frequency P is at 0.5, Q is then of course also 0.5. And if you start with P equals 0.5 and let this population of 9 individuals just randomly breed with one another for 20 generations, this is what happens to the frequency of P. These blue lines bounce all over the place. Each of these blue lines represents a different simulated population of 9 individuals. When those 9 individuals breed with one another, there's a chance that in the next generation, when they create 9 more offspring, that the allele frequency for P has gone up a little bit or gone down a little bit. And you'll notice that in a number of these, the lines actually terminate at the top or the bottom of the graph, meaning that the frequency of P reached 100%, or 0, totally gone. So there are only 1, 2, 3, 4, 5, 6, 7, 8 of these simulations in which the frequency of P and Q, there's still both of the alleles in the population. In 5 of these simulations, the frequency of P crashed to 0, it's gone. In 3, 4, 5, 6, and 7 of these simulations, the frequency of P went to 1, and that necessarily means that the frequency of Q is 0. And there are a bunch of different simulations here of the same principle, just to show that when it's random, the frequency can go any direction. It can jump by large leaps, or it can change by very small amounts. It's all very random. But what I want you to get from our discussion of genetic drift is 2 things. One, it's completely random, okay? So the allele is just as likely to reach a frequency of 1 as it is to reach a frequency of 0. And the other thing is, look how long it took for, 7 plus 5, for 12 of these simulations to reach a frequency of 1 or 0. They became fixed for P or Q. But instead of 9 individuals, if we have 50 individuals in the population, look what happens over 20 generations. It's a very different picture. The time that it takes for one allele to either drift randomly to 0 or to 1 is heavily impacted by population size. The more individuals there are in the population, the slower the population will change allele frequency just by chance. Okay? I do want you to know that in genetic drift, one of the alleles absolutely will become fixed. No matter what, it's going to happen. It could take a long time, but it is absolutely going to happen. You can see on the graph on the right that each of those lines is either kind of trending up or trending down, and that will give you some idea of the probability of it reaching a frequency of 1 or 0. But it's going to happen much more slowly in a population with 50 individuals than with 9. And this is why conservation measures are implemented to protect species that have very low numbers. If a population has very low numbers, genetic drift becomes a very powerful mechanism of change, and you can even lose very beneficial mutations. Most of these things that are drifting aren't good or aren't bad. It's just changing by chance. But drift can act much more strongly in a small population, and it can really detrimentally affect a small population. So the genetic variation in populations is lost to fixation. If one of these alleles is going to become fixed, reaching a frequency of 1 or 0, then we are definitely losing allelic diversity because the other allele disappeared. Okay? And so we gradually lose variation to fixation, but remember we are constantly gaining new variation through the process of mutation. And the last thing I'd like to point out is that at any time, the probability that a neutral allele, one that, let's say a silent mutation, doesn't affect the phenotype, the frequency of that allele is equal to the probability that it will drift to fixation. So you can see as those lines on the left started going down, they got pulled faster down to a frequency of 0. Okay? The lower the frequency of the allele, that is the probability that it's going to be retained. So it gets lower and lower and lower. It goes from a 50% frequency to a 25% to a 10% frequency. Then we're down to a 10% probability that it's going to be fixed, reach a frequency of 1. The lower the frequency is, the more likely it is to disappear, and vice versa. Okay, so that is an overview of the principle of genetic drift, an important principle that is covered in great depth in an evolution class, but in this class I simply need you to know that allele frequencies can change by random chance, and they do that in ways that are scaled to population size. Now, when populations become small, we run into an issue that you've certainly heard of, and this is inbreeding. Inbreeding is problematic because we have an increased chance of individuals receiving deleterious alleles, 2 copies of them. They could be homozygous for something that is bad. That was very rare in the population, but because they are the product of inbreeding, right? Let's say in this case, their parents were cousins, okay? If their parents were cousins, then they are likely to receive this recessive allele that was in their great-grandparent, simply because that allele is much more common in their family history than it is in the broader population. And so you can see this homozygosity for deleterious alleles become much more common in populations where there is lots of inbreeding. Okay, so when you think of inbreeding, you probably think of people, but as a biologist, as a scientist, you should think about it in the context of when population numbers become small, then individuals are reproducing with others that are very closely related to them, and you end up with all sorts of genetic difficulties because of this increased level of homozygosity, okay, especially for things that are deleterious. This is why you don't breed brother and sister puppies, right, or dogs or cats, okay, because the consequences could be pretty dire. Okay, so how does genetics play into this process of the origination of different species? Okay, why are we talking about biodiversity in the context of genetics? Well, it has everything to do with how continuous populations are genetically. Are they exchanging alleles with one another? If they're freely exchanging alleles, then they're going to be the same thing, okay? Your dog and your cat are not going to exchange alleles. They're not going to mate with one another, so the cat alleles and the dog alleles are maintained separately. And so this allows them to accumulate mutations over time that make them more and more different. At the very beginning, when speciation happens, the two different lineages that are becoming different are probably not all that different, but those differences accumulate rapidly over time through the process of mutation and through the absence of genetic exchange between the species. So what makes things different species? You look at a dog and a cat and you know that they're different species, but what is it that makes them different species? At the genetic level, it's this failure to exchange alleles that I've mentioned. At a biological level, we think about why these populations are not exchanging alleles in the context of these different mechanisms that isolate them from one another, okay? So there are mechanisms that prevent organisms from creating a zygote, right? Your dog and your cat are not going to get sperm and egg in the same place and create a zygote. It doesn't happen. So the isolation that prevents them from mating with one another is a pre-zygotic mechanism of isolation, okay? The differences between Indian elephants and African elephants, the reason that they don't exchange alleles isn't that they couldn't physically do it or they wouldn't find one another attractive necessarily. It's that they are in different geographic regions. So if you go through and think about these different reproductive isolating mechanisms that prevent alleles from moving from one population to the next, if it is effective, that means those two populations are not going to be the same species, even if they are right now. So you can have populations that are separated geographically. You could have them separated seasonally, okay? There are different species here of frog in Mississippi that breed at different times of year. So two frog species, one that breeds in January, the other that breeds in July, those two species are not going to ever mate with one another, okay? There are behavioral differences. This could be something like the songs that birds sing. When you hear a bird singing, it might be doing that to attract a mate, but it's only going to be attractive to another member of the opposite sex of that same species. There are also mechanically isolating mechanisms that prevent organisms from creating a zygote. You can imagine a wolf and a mouse cannot create offspring because they are physically incapable of reproducing, okay? And then there are physiological prezygotic mechanisms whereby the gametes just fail to survive, right? If you put a gamete from species A into the oviduct, the female reproductive space of a different species, if the chemistries aren't properly aligned and compatible, then that sperm might just basically be dissolved or expelled. So the postzygotic mechanisms, these are mechanisms that keep populations isolated from one another allelically, despite the fact that individuals of each species might come together and mate and produce offspring, but their offspring might be weak or just non-viable. They might be sterile. This is the case in horses and donkeys. When you breed those, you get a mule. Mules are sterile. And so the gene pools of horses and donkeys are actually kept apart. There's no mixing of alleles, even though you can create these mules. There's no further introgression or genetic incursion into either species. We can also have F2 breakdown, whereas F1s might be good, but F2s are weak or sterile. So if I give you a mechanism, if I explain a scenario whereby two organisms are isolated from one another genetically, you should be able to tell me whether it is a prezygotic or postzygotic mechanism that is isolating them. Okay. Now, as these populations change over time, the x-axis here, there may be changes within the lineage. So there could be a stasis, a long period of no change. And we certainly see species that live on this planet today. An alligator is a great example, in which alligators have not changed much in a long time. Okay, there's been lots of periods of stasis. There can also be periods of anagenesis where a species changes quite dramatically. So you can see from where it says species 1 to species 2, they get those spikes on their back. That is change over time without there being multiple species created. But when we go from species 2 to 3 and 4, this is speciation. This is when we get one species becomes 2. All right? And these are species 3 and species 4 are indicated here as having different phenotypic characteristics. They wouldn't mate with one another because it looks like they live in different areas, and they probably behave in different ways when they're trying to attract mates, so they don't mate with one another. This process is the combination of time and change in form and phenotype that results in the isolation and the origin of new species. Okay, the text in the region of the text where they talk about speciation covers a couple of examples. This is one, a species of snapping shrimp that lives in the ocean around Panama. But what's interesting is, if you think about Panama, here's Panama. Here's a zoom of Panama. This is the land bridge that connects North and South America. Okay, it separates the Pacific Ocean from the Caribbean. What's interesting is 2 million years ago, wasn't here. Okay, 2 to 3 million years ago is when this Panamanian Isthmus formed. It created a physical connection between the land masses, but what it also did was create a physical separation of all the critters that were living in this area. Some were now stuck in the Caribbean, some stuck in the Pacific. And over that millions of years time period since then, when we look at species like the snapping shrimp I just showed you, there is one species on the north here in the Caribbean, and there's one species in the Pacific, and they're each other's closest relatives. They look very similar, and over the past few million years, they've been physically isolated from one another and diversified, became different from one another. Pretty remarkable. So speciation can happen over very long time periods, or there are also instances where, as in the Great Lakes of Africa, the cichlid species here have speciated so incredibly rapidly that within 100,000 years, they have diversified into hundreds of species. So whereas the shrimp take a couple million years to go from 1 to 2, this lineage has taken 100,000 years to go from one species to 300 or 400, depending on the lake. Okay, so there are really interesting patterns of diversification of species, and there are many things that can lead to the differences that make all of these cichlids, even though they're closely related to one another, genetically very similar, there are many things that keep them from reproducing with one another. Part of it is they eat different things. They have different colors that they use to recognize each other as mates. They live in different habitats. They lay eggs in different places. All of this has happened so rapidly in these lakes that were empty of fish until these cichlids got there. So they were able to do lots of different things and take over lots of space very rapidly. I'm not going to cover this example in great detail because it's one that is more for an evolution class, but there is a little section here that in your text that talks about the rate of evolution and speciation. So evolution can be slow or it can be incredibly rapid. Okay, it all depends on the strength of selection. Now, I want to finish off here talking about the complex origins of the human genome and studies that are ongoing. I've already mentioned this particular figure and how we are able to use archaic DNA or ancient DNA to understand the evolution of our species, but also consider the evolution of our species with respect to other hominids that lived in the past million years. So a recent discovery in Russia in a cave found what was not another caveman or Neanderthal, as you would commonly call them, but they found this finger bone, just a finger bone. Okay, they had other elements of the skeleton, but from a small pinky finger bone, they were able to extract DNA and sequence it, and they thought it was a Neanderthal or a human. Turns out it was neither, it's a third species. So they sequenced the genomes of these Denisovans. Okay, this is one of the sequences that you look at in your lab. And when they sequence these, they find them to be incredibly different. So they're a much earlier lineage of hominid, very different from humans and Neanderthals. And I just want to give you an inkling of how different they are. So I want you to look at this slide first, in which we see a phylogenetic tree showing modern humans being the most closely related to Neanderthals. And we have a common ancestor, and then you trace that line back to a common ancestor with this Denisova hominin, okay? Going much further back, not hundreds of thousands of years, but 1 million years back to the common ancestor with that species. Now let's look at this plot that compares the genetic differences between these species. So this is looking at the mitochondrial genome of humans, Neanderthals, and Denisovans, okay? And on the x-axis is how many differences are there when you compare these, okay? So on the left, the far left, you see there are two spikes, two peaks there for human. If you compare two people from the same people group, let's say two people from China or two people from Peru, then you are going to see that on average they have somewhere in the neighborhood of 40 to 50 differences. Do you see that peak for that portion of the graph? Now, if you compare two humans that are from different people groups, let's say somebody from China and somebody from Senegal in Africa, then you might have an average of 90 to 100 differences between them. That's what explains those two different peaks there, okay? But if you compare a human to a Neanderthal, on average, you are going to see 200 nucleotide differences, 200 mutations in their mitochondrial genome. Remember, this is only out of about 25,000 nucleotides in the mitochondrial genome. If you compare a human to a Denisovan, there are nearly 400 differences between humans and Denisovans. So you can see that we are, of course, the same species as other humans that are around today. We are somewhat different from Neanderthals, but pretty similar. But we are quite different from Denisovans, far more different from Denisovans than we are Neanderthals. So a really interesting application of this paleogenomics. And when you, if you get, a personal genetic test and run, say, 23andMe or Ancestry genetic test, they will tell you about what proportion of your genome is Neanderthal, which is pretty remarkable because there are a number of alleles in humans that were most common in Neanderthals and presumably originated in Neanderthals. The only reason they exist in humans today is because they originated somewhere around 706,000 years ago, let's say, or somewhere between there and 370,000 years ago in Neanderthals, but the populations were still mixing a bit before 370 million years ago. So, all right, that's it. That wraps up chapter 21.